Um, all right, hello everyone. Welcome to MedChem yet again. Yeah, I'm not, I guess, to address the not feeling so good. Yeah, not feeling so good at the moment, but I didn't want us to get behind it all, and I'm sure you guys are eager to learn some more medicinal chemistry, and I'm eager to teach you. So we're gonna talk about that a little bit, sort of some things having to do with um, lead discovery and optimization. We're going to get sort of conversation started in that area. I'll keep you guys posted in the next day or two as to how things are going on my end. We may or may not have another another Zoom focused one on Friday, but let me see how I'm feeling tomorrow. But I hope you guys are all feeling great, and we'll get talking about some new content. I did want to mention, it sounds like you guys could see that your quiz one is graded, so that's good news. And across the board, you guys did really great on that. I think there was a lot of good reflection on sort of what's going on, right, with COVID-19 and etc. So I was really pleased with that. And we're still cruising through exams. You guys are doing pretty well, I would say. So I'm looking forward to sort of sharing, how well you guys did very soon. But I'm going to go ahead and get sharing the screen and getting into our discussion for today. Minimize that. Yeah, so now we really start getting into so a lot of the fun stuff. Just want to make sure how are the exams looking so far? They're looking pretty good. I think you guys might beat the average from last time, which will earn you donuts. We'll talk about that once you guys get them back. Okay. Yeah, so we're going to talk a little bit about today. We're just going to sort of introduce a little bit of information having to do with lead identification and optimization. And sort of, you see sort of this like introductory pinwheel here that sort of, talks about sort of each of the, sort of the components affiliated with sort of identifying an issue and then creating sort of a hypothesis for addressing that issue. And so, so you can see, right, there's sort of a design phase, there's a synthesize phase, test phase, and analyze phase. And we'll touch a little bit on sort of each of those as we go through. A lot of the content actually at the beginning of this is going to be a little bit of sort of a little bit more in-depth conversation about what you saw sort of in the introductory slides when we got started in section 1. And so to get our conversation started, one of the questions that I always had, in thinking about sort of lead identification, what do they go after? How do you decide what to go after? And usually, right? So how do pharmaceutical companies decide which disease to target? Usually that's driven by economic factors, right? So if they're going to make a very large investment, right, it has to pay off for them, right? They have to have that return on investment, right, and then be able to continue to make money from that, right? So diseases present in the developed world are typically targeted primarily, right? So you see a lot of sort of heart related things, right? Breathing, right? Lungs, and then of course, right? So there's other key ones. So for example, like diabetes, right, is huge. And so, sorry, I met a couple people. But I think it's important to note, right, that the research on tropical diseases. In progress. Okay. Research on tropical diseases is mostly done in partnership with a sort of a series of sort of sort of non-profit type organizations that I'm sure you've heard of. If you haven't heard of Wellcome, that's a really, really great one to look into. Bill and Melinda Gates Foundation, of course, is a huge one. I'm sure that's one you've heard of. And one I like to highlight is actually the Chan Zuckerberg Initiative, where they, I guess at the beginning of it, it was sort of, they wanted to sort of, come to a cure, right, for all disease in a certain span of time. And that span of time has changed since then. But I think, it seems like almost like, sort of a pretty lofty goal. But to think sort of you'd come up with a good way to cure disease over any really period of time is really great. So I really encourage you to go take a peek at that and sort of all these websites to see sort of all the initiatives that they're part of. Yeah, so one of the key aspects to this is going to be choosing a drug target. After the therapeutic area is identified, right, it's time to identify a suitable drug target. And so, if you look at the pie chart down here, or you'll see that, enzymes are obviously a really, really huge target, right? And then you sort of have the G protein coupled receptors, sort of sitting in the 2000 range, ion channels, and then nuclear hormone receptors. And oftentimes, right, this is related to sort of their accessibility in vitro, right? So being able to isolate them, be able to actually interrogate whether or not you have binding, or if that binding is inducing sort of a biological response. And so enzymes, I think are going to sort of take the cake for quite some time. But of course, G protein coupled receptors are so, important, right, and sort of signal transduction, that they're of course going to be a key player as well. Right. So, right, the caspases are a really great, sort of example to maybe begin with and sort of get into this discussion, right? So, there's enzymatic protease drug target, right? So proteases, they're going to, typically cleave amide bonds, right? And we talked about that a little bit before. They catalyze the hydrolysis of important cellular proteins that play a role in inflammation and cell death. So those involved in DNA repair and regulation, right? So they're really these enzymes that are capable of just like really tearing apart proteins. I think that's, that's really powerful if you sort of, think about that sort of on a molecular level. But in particular, right, they recognize aspartate groups within the protein substrates. So it is it's a really great area for molecular design. So you can see, right, if you have something that targets aspartate, for example, where you see this sort of aspartic acid here, right? So if you want to inhibit that process, you usually create something that's going to mimic, right, the aspartic acid. And so you have, I think, a maybe more blatant example here. So a mimic for aspartic acid. This is essentially aspartic acid with sort of, a molecule attached to it. But then on top of that, right, you have these situations where you can sort of develop sort of more cyclic structures, almost in sort of a rigidification type strategy, to potentially, create something that's more important. But most importantly, right, these are both mimics for aspartic acid, right? And they sort of are, they function as sort of an inhibitor, right, for the caspases. I think it's really cool. Yeah, right. So if a drug or poison produces a biological effect, right, there must be a molecular target, right? I think that's a key sort of design principle in medicinal chemistry, right? So I'd ask you to sort of recall, right, the example of nitric oxide as a chemical messenger from module 2. That's sort of a really nice example of this type of process, right? And so oftentimes, right, a great starting point are the body's own chemical messengers, right? So, the design principles sometimes are, we really want to get to something that we already know binds, right? And so these are really two really great starting points, right? They look pretty complex, but I think it's important that, especially, I think you have an appreciation for this after maybe a certain exam question that you had, that a lot of these are accessible via sort of simple peptide couplings, and that's sort of been a transformative type of transformation for accessing compounds like this. But some examples, for what that, kind of what that translates from into, right, is so, you have these endogenous ligands, right, that you already, looked at, you hopefully recognize in a matching scenario. But then what they can do is, you can sort of map those on to the creation of the marketed drugs that are seen here, right? So you can see sort of the serotonin structure, right, is present in this molecule here, right? And so the same with acetylcholine, right? You have sort of this sort of charged component of it, and sort of this one is also charged, but also looks very much like sort of the geometry about sort of that nitrogen, right? It's really, really cool. And then of course the norepinephrine, has its, has its sort of marketed drug affiliated with that as well, right? But sort of we're gonna talk a bit about sort of it doesn't seem, you're like yeah, Kyle, well, this doesn't seem obvious, the jump from norepinephrine to this compound here, but we're going to talk a little bit about sort of those design principles probably sometime next class. Right, so we talked a little bit about target specificity, right? That ends up playing a huge role, right, in sort of drug design, right? The more selective a drug target, the lower probability of having undesirable effects, right? So between species is of course a really great one. We talked about this. So choosing targets that are unique to the microbe or species you're targeting would be ideal, right? Because it hopefully will not affect you, right, your own body. But then within the body, right, you want to aim for the highest type of selectivity that you can. So we talked a little bit about sort of these CYP3As, right? Those cytochrome P450s. And those ones, almost every molecule that you put into your body comes in contact with those. So you can imagine that, selectively targeting, if you want to selectively target, for example, CYP3A, that's going to be a bit challenging, right? But it gets easier, right, to sort of target a specific enzyme, right? It's just that you don't have sort of unwanted, sort of effects in the body, sort of undesired complications, right, with the drug, right? If you can start to target ones that that have sort of a larger specificity, like CYP2D6 would be, right, some of the targets that would be really great, right, because it's really honing in on that, and it's not going to bind to all the CYP3As that you have in your body. So yeah, right, so this often occurs by nature of selective targeting in the enzyme or receptor. Really good example, right, is sort of in sort of the live nerve, right? So you want to be able to design, let's say, chemical messengers, right, that are going to selectively bind to the receptors that you're targeting, right? So this is sort of just a really good example where this definitely comes into play. Right, so possible pitfalls, right, for choosing a particular target, right? While the target may seem correct, right, the process in the body is going to be complex, right? And I think we have at least a working appreciation for that now. So sometimes, right, more than one target may need to be addressed. And oftentimes a decrease in efficacy could mean that a blockade event has occurred, right? So if you have this thing, they describe it as a town center, right? That's the location you want the drug to get to. But then for some reason, right, that ends up getting blocked off, right? And so your efficacy, right, goes down because it's not as efficient of a process in getting to that location. But I think it's important to note that if it can't get to that location, it has sort of other options to get to that location. Although you're going to see a bit of a decrease in efficacy, as well as sort of undesired side effects. Another, a key area that sort of, come up, I would say over the course of the last, 20-ish years now, is sort of this concept of polypharmacology, right? The whole idea was to design, molecules that could target sort of multiple different targets, right? So hitting a number of different targets selectively. And sort of as an alternative to combination therapy, where you take, let's say, two molecules that are going to, sort of have sort of that combined effect, biological effect on the body. The disadvantage with the combination therapies, right, is the number of different medications associated with the dose regimens, right? So we talked a little bit about sort of staying in that therapeutic window. And, of course, for every molecule that you take in, right, your therapeutic window for each of those is going to be affected by sort of a range of processes in the body, right? So by having one in which you can sort of really sort of understand what the therapeutic window of that is and only have to regulate that would be ideal, right? So these dual and triple action drug discovery is now, of course, a growing research area. But the thing I would say, this sounds like really great, right? It's sort of the onset, but I think the important aspect of that is that, if you have something that can target let's say 2 proteins selectively, you also have a situation where it might also target other proteins selectively that are not ideal for your current situation. So I would say the design of these is really when they work, right, they're a real home run. But I would say that they're still a bit challenging to get after. And actually, one of the medicinal chemists that we're going to have in Medicinal Chemist Week actually works, I think, pretty heavily in the polypharmacology area. So I'll have her speak to that a little bit. Yeah, right. So identifying a bioassay, right? As you guys might imagine, if you're thinking about sort of screening, thousands of molecules, right? You want to have something that, let's say you screen one of them, right? It doesn't take like a week, for example, to find out the result. And so, one of the big things that's come about, I would say, in the last 20 years, right, is so this high throughput type of experimentation. But this is actually sort of a coin phrase that's used, pretty heavily in medicinal chemistry, and that's fail fast and fail cheap, right? So you want to come up with something that's sort of quick and dirty, right? That can tell you, whether or not, a drug is effective. It is one of the most important parts to a drug research program, right? For being able to do this in high throughput. And then you want to come up with very simple, quick, and relevant bioassays to do this with. Most testing is done right in vitro. So this goes back to sort of that discussion on the enzyme, right? That you need to have a target that's readily accessible, and that makes not only for sort of accessibility, but also for just being able to sort of locate it and purchase it from a vendor, for example. And so, like, if you follow around this pinwheel, right? So a couple of notes to mention, right? These automations require a ton of sort of robotics. And we'll talk a little bit about that as we go through. High throughput, you're looking at screening somewhere in the neighborhood of 1 million data points a month. I would say right now that's maybe wildly outdated, right? You're looking at somewhere pretty close to 1 billion a month. Now it's getting the bar is getting really high. Right, you want to come up with high throughput screening readouts that are really efficient, right? So ones that sort of based on fluorescence and things. Assays for any types of targets is also really important, and oftentimes in like a single well, you want to come up with a way that you can test hundreds of compounds. And so we'll talk a little bit about that as we go through. So, right, high throughput screening is critical, right, in sort of development of a drug. And so this automated testing of a large number of compounds versus a large number of targets. You might be asking, okay, so where do they get all these compounds? So what I would say is sort of there's sort of several different locations that sort of keep, right, libraries of compounds, and then each pharmaceutical company also keeps their own set, and so that they can exchange, right, amongst companies too, which I think is really cool, right? So med chemists are really hard at work making a whole bunch of compounds, and they can be shared, right, sort of across pharmaceutical companies. Usually they're going after sort of like, there is a common cause, right? They're not all competing all the time. Right, it should produce an easily detectable effect. So radio labeling is huge, characteristic signatures, etc. And so positive hits usually bind in the micro to nanomolar range, right? So I think it's really important, not only to note that you need binding, let's say, to the target, and it needs to be effective, but the dosage of the compound that you add can't be remarkably high, right? That's not going to be a great starting point for you, because sort of on the other end of things, right, you have to come up with a way to produce a ton of that compound, right, in the case that your dosage is high. Not to mention, right, sort of ADME related items. And so this is kind of what a screen looks like, right? You have sort of small molecules or these RNA type molecules, right? You combine those in a plate. Usually these are about 1,500 wells or so, with a particular target that you're interested in. And you create sort of a radio labeling or a fluorescence assay that's going to give you that information. So you have sort of plate readers that can take a snapshot of those, the whole thing. and see all 1500 at the same time. You have high content screens where you can monitor sort of what the cells are actually looking like to get a sort of an understanding of sort of what's going on. And then you have sort of this kinetic imaging screens. That's very similar, I think, to the plate reader. But sort of once again, right, it shows you sort of like the literal cell morphology, sort of, the actual kinetics of the process in each well, which I think is really neat. And that's sort of automated now. So that's something that, I was really sort of aware of, several years ago. I think that was really, really cool. For anyone that's familiar, I think in organic chemistry, you guys probably covered nuclear magnetic resonance or NMR. Another good way to monitor whether or not you're getting binding to a substrate is to look for sort of modification of relaxation time in the NMR. And so, for example, if a small molecule typically in the NMR looks sort of like this peak here, when it's bound, it's actually going to shorten the relaxation time of that proton, so that H atom. And so you're going to see a change in that particular hydrogen, for example. And then sort of you use a host of sort of validation techniques that you definitely don't have to know, but you're just noting that NMR is sort of a key sort of contributor. This is a huge one, and there's also high throughput ways to do this. It's really neat. Screening based on the affinity for a target is another big one, right? So for example, if you have a pool of compounds, this is exactly what I was talking about before. You have a ton of compounds you need to screen, but then you have a fluorescent ligand target complex, right? So these things all glow at the beginning. And so what you actually look for is that ligand to be, sort of dislodged from that small molecule, to be dislodged from the active site of the binding site. And then another molecule to bind, right? So if you see sort of those, I guess, for lack of better words, the light turn off, right? Then you know that you have a hit. And then from there, there's nice sort of tagging options to take that out of the well, and then you can identify what that small molecule is. So surface plasmon is another key way to monitor binding. I would say it's a little less popular now with a lot of the techniques that have come about in recent years. But ultimately, what you do is you sort of bind the ligand or the molecule that could maybe bind sort of several different targets to sort of a it's almost like a glass plate, and then you flow targets over that. And then if there's a binding event, you're shining, light on that plate, and you start to see sort of changes in the refractive index. And so that's actually sort of monitored on a computer. It sort of shows up as like small waves, which I think is, really neat. I would say a less popular one, but it definitely was really popular. I would say even like 10 years ago. Detection of ligands that are bound to the target can also be, you can also use sort of these readouts based on radioactivity to light. So you start with these scintillation proximity beads, these SPA beads, and basically you attach via, you don't really need to know this information, but biotin biotin's sort of a small molecule that's going to bind to, let's say, a protein or the bead, and that molecule is actually attached to a certain type of receptor, so your target. And so you flood it with a bunch of molecules or a particular molecule, and basically then you shine light on it, and you see whether or not they glow, and that glow is happening from sort of an energy transfer from sort of a radio labeled ligand or molecule to the SPA bead, and at that point, those beads, oftentimes they're even magnetic. You can pull them directly out of the solution, and you can identify sort of what molecule bound, for example. Isothermal titration calorimetry is another really interesting technique. This is used, typically for, when you want to really understand sort of the binding affinity of a particular ligand. And when I say ligand, again, these are sort of the small molecules or, potential drugs. And so, it basically monitors the temperature fluctuation as you add more and more of the compound. And you could start to, so basically inside of here, inside of the cell, you'll have sort of the target, and you'll be sort of syringing in some of the ligand, and you'll see sort of these responses that are occurring sort of in the middle. And then based on those responses, you have sort of like, you have a sort of a graph of the concentration versus the response. And you're able to monitor to actually find what sort of a KD or dissociation constant would be for the molecule. Really, really neat. I think this is used a little more downstream in the workflow, and when you really want to start to understand the binding affinity of the compound. Yeah. So another one that's really huge, right, is this virtual screening, right? This is in fact, there's a ton of people that believe that, this is the way to go all day. And I think eventually this is going to be, maybe our primary, sort of, route for screening. So basically, right, they can set sort of a series of parameters up in a computer, right? And they can start to say, okay, make theoretical molecules. And so they'll create, so for example, we'll give sort of a lead compound, and we'll say, okay, put methyls on it all over the place, or put one methyl here, and it'll do that at random. And it'll screen those against the target. And so what you end up with is sort of a readout. Usually these are, I would say week-long experiments, but it can look at, somewhere in the neighborhood of, a couple billion molecules maybe per week. And it'll start to identify molecules that may be of use. One of the challenges though, right, is it's going to come up, with sort of molecules at random. And sometimes those molecules are just not, they're almost, they wouldn't be, they're likely not going to be a great option just from sort of their, from disobeying sort of Baldwin rules, for example. And oftentimes those will get sort of shuttled out of the consideration. But for ones that it creates that are reasonable, those are then made in the lab and tested, right, in vitro. Just want to give you a good, a feel for what things look like. You absolutely do not need to know any of these steps. If you do, I mean, congratulations, it's pretty great. But oftentimes what we'll do, sort of from a medicinal chemist standpoint, so the synthetic end, is we'll start with sort of a molecule. This is sort of in creating a library of molecules. You start with a molecule, you attach it to a bead, and that bead is sort of what you use sort of for purification, right? So you'll take, for example, this molecule got attached to a bead right here, and then you can start to do the subsequent transformations. And you can imagine from organic chemistry, right, when you get all that mess in the flask, you're like, I'm not sure what to do with that. There's really good ways to sort of wash bead in the molecule, so you end up sort of purifying the molecule that way. You can imagine, right, these can be combined then to make sort of a whole bunch of molecules. So this is usually like considered sort of an on bead type synthesis, and you can do that one at a time, or you can do this multiple times, and we'll talk about that in a few slides. Right, so finding the lead compound, right? Natural products are really, really great location, right? They have a plus side and a minus side, right? The plus side being that they're usually pretty good at what they do, right? I would say the minus side, or the more challenging side, is that sort of functionalization of them is not particularly easy. But if they do well sort of on their own, right, we come up with good ways to go ahead and modify their structures. And yeah, so this is sort of, similar to the chemical messengers, right? You have this sort of situation where you have the natural product on the left, and then marketed drugs on the right. And so you might say, hey, you know what, you just told me it's really challenging to go ahead and modify these molecules. And I would absolutely tell you that indeed is the truth. But what you can see is, right, it's not, for example, this top one's not modified a ton. Really what they've done is they've taken off, right, this sort of acyl group in the top right, and then they install sort of a new acyl group. And those are, you'll often find, right, that the marketed drug is something that was just accessible from synthetic organic chemistry. And so, but that's sort of a whole different type of course to teach. But ultimately, right, usually there's not a ton of modification to natural products, and usually the natural products are used on their own if they're going to be used. Right, microbes, right, are another really great source of drugs, right? So these are ones that sort of come from them, right? Nice representative examples. And again, these are going to be good, right? In that they, these hopefully they won't affect our body as much, and hopefully they'll be able to target the microbes maybe selectively. This is an inspiring figure, and I know it's fuzzy, and I've been trying to make it not fuzzy for now a couple of years and haven't been able to do it, but this is super cool, right? So if you think about, this is like my punchline slide for natural products. I'm a huge fan of natural products, if you haven't figured that out yet. But look at this, right? You have, on the right side, start there, right? The number of approved drugs, right? This features all the approved drugs as of 2018. And then if you look at the left side, right, marine natural products. I mean, look how much that overlaps. That's so fascinating to me. So natural products for a long time were, especially when we didn't have sort of synthetic methods to get after, really complex molecules. This may be a product of that, but they were sort of like the inspiration for drug candidates. And so, I would say there's large advocates right now still that say, the best starting point for a drug is definitely a natural product. I can go off on that for several hours, but I'll spare you that. Antibacterials, right, from animals, right? We can, from the frog here, right, are another really great source of drug. Right, so from venoms and toxins, right? So you can imagine from those, you're going to get sort of these antihypertensives. We talked a little bit about that before, and muscle spasm prevention, right? So, of course, you can imagine that, making these in the lab, that sounds like really exciting. Bring a student in and say, you know what, I really want to get after a really nice synthesis of tetrodotoxin. It's a super great idea. But the thing is, I probably wouldn't have anybody do that, because in the case that they're exposed to it, they're going to have some serious problems in the lab. But nevertheless, there are groups that do work on those and come up with really cool ways to make them. Right. A lot of medicines, right? You probably can have an appreciation for this, right? Comes from the result of a placebo effect, right? So willing a drug to work, right? So you take something and you're like, oh, I feel really good. And so, but it may not actually be causing the effect that you're thinking it does. It might have just been time, right? And your body is taking care of, whatever it needed to. So, I would say keep a lookout for the placebo effect. You're not gonna know a ton about that. Might be a nice multiple choice question, but nevertheless, I think it's worth noting. Yeah, so this has been a huge area as well. Thinking about sort of, what are the inspirations for lead compounds? You would think, right, let's say I'm going to do like a 10-step synthesis to get to a biologically active compound. It would be a great idea that, as the molecule, as you continue through the synthesis, it's going to become more and more sort of drug-like, right? So one key area is now every intermediate that the pharmaceuticals companies make, they actually submit those intermediates to screening, right, for across a number of different targets, but definitely sort of on the target that they're after as well. There's two important sort of aspects to that. One, you might identify a new drug, and number two, if let's say 5 steps in of a 10-step route, it does essentially the same thing that the 10-step route compound does. Think about the amount of money that you save from that component of it. That's really exciting, sort of a really exciting space. It really didn't dawn on them for quite some time. I would say 2005, I would say, or so. This sort of became a really big area. Really neat. So there's actually places that literally are just like these, you just deposit compounds and they'll actually screen them for free. So that's something that our lab definitely also contributes to, which is really neat. Yeah, so the Me Too drugs, right? I try and tell you, stay away from those. And yeah, some Me Too drugs, have made it, right? But you could see sort of, based on Captopril here, right? This is maybe different enough by this Hoffman LaRoche molecule. But then, when you jump from, even something from lisinopril here from Merck to enalapril, right, also from Merck, this is usually not the jump you usually want to make, but they did successfully. So it's not to say that it never is a great pursuit, but it wouldn't be like the number one, I think, candidate for you. Right. So excellent starting point for optimization, or going to be these sort of modulator type molecules. And so, you might start actually with, for example, right, Viagra has a ton of sort of studies on that, where I think they've functionalized maybe every part of this molecule to sort of turn on new types of sort of implications for disease, for example. But especially these molecules that sort of contain sort of a lot of like aromatic type moieties, right? Those are easy to functionalize, based on sort of what's known sort of on the synthetic end of things. So those end up being really easy to access, for example. Yeah, so this is what I was talking about before, sort of in that synthesis component to accessing lead compounds. You have sort of these two different camps of thought. One of them being in this combinatorial synthesis, right, where you split. It's called the split synthesis method. So each well or reaction vessel contains many types of support bound molecules. So those are those ones on the bead. And then you can perform sort of the same type of reaction on all of them. And so they all move sort of as a unit, sort of to the next stage. So in the end, right, each well, you have a whole bunch of molecules in each well, and then those can be tested against the target. Alternatively, right, you have parallel synthesis, and that's one bead, one compound, right? So you're really going after sort of that one compound, but you're sort of on each bead, right? There's a different compound, so you're taking through the sort of very similar type of step, and you get less molecules out of that. But usually, this is sort of comes in when there's sort of a more informed target that you want to get after. Right, fragment-based screening has become huge. So where you have the situation where you have high throughput screening, where we talked about before, where sort of you screen sort of a diverse library of compounds, and you're able to sort of through a biochemical assay, you're able to identify a hit or something that binds. But what people have started to do is actually look for binding of fragments of molecules. And so what they do is they mix sort of A and B. So this is in the B column here, right? They mix A or B into the bioassay, and they look for binding to certain sections of the molecule or certain sections of the macromolecular target. And then what they'll do is at the end, they'll link them. So you sort of A and B on their own are not going to produce a biological effect, but when they're linked, they might, right? And so that's something that they'll also pursue. And then you have sort of this dynamic template-assisted drug discovery, right, where you have this dynamic, right, fragment ligation, they call it. So you have something that's really tightly bound a fragment that's really tightly bound to the macromolecular target. And then you subject that to sort of a series of other possible fragments, and you look for binding that way. Right. So one of the, this whole area, I would say, is, considered now called epitope mapping. So you have this epitope, right? So it's a small molecule that binds to a region, but has no activity on its own. And then they can be combined to potentially design a drug, and that's generally called epitope mapping. You're mapping out the active site of the protein, right? So you have the screen for the first ligand, and then you sort of optimize, that molecule for binding. And then once you have that, you screen for the second ligand, while the other one's bound. Because again, right, there's an inductive effect that occurs. So you need to sort of look for the second one after you've modified the first one, or I guess you optimize the first one. And then ultimately, right, you link them. So and then you sort of look right for the biological response. So you might say, Kyle, well, how in the world do you link them? Okay, so one option, right, is this sort of fragment evolution where you have sort of A, right, has sort of the first target, and then you put in B, and that has sort of an appendage on it that can link up to one. And then in B, right, you have exactly so what we mentioned before, right? You have you find the binding of the first thing, binding of the second thing, and then in the end you link them. Finally, in C, you have this fragment self-assembly, right? So you attach something to 1 and 2 that ultimately, for example, people might use DNA as sort of appendages off of those, so those sort of link naturally just by binding to the macromolecular target. Yeah, one of the key strategies for doing that linkage, and you definitely start this slide, is using the click reaction. I'm sure this is one that some of you have definitely heard of, and it provides a great in situ look at bringing fragments together. This is arguably one of the most important discoveries sort of at the sort of chemistry biology interface. This is super biocompatible. It's used as a linking strategy for all sorts of things, to link things to proteins, to link molecules together like this, to link proteins to DNA, etc. It's a really important reaction to know. I wanted to wrap up just by sort of giving you sort of a snapshot of what things look like. If you go out to, if you want to sort of bring in a compound library, there's a ton of them available, and they're for all different things, right? Anti-infection, ion channel compounds, epigenetics, GPCRs, etc. Right? You can type that in. You probably Google it, and you'll find it. This is just on one website. Sigma Aldrich sells all these. They sell all these, sell all these ones. And well, there's probably a third page, but nevertheless, right? You can sort of find a whole bunch of them. Really, really easy access now. They're a bit expensive, but also you can also reach right out to a pharmaceutical company. For example, if you're in a lab, you can reach out to the pharmaceutical company. They'll usually give them to you. And on that note, I wanted to sort of have that introduction, and then we'll get into sort of optimizing drug interactions next time. That's where it starts to get really fun. You guys are really, I say that every class. I mean, it's always really fun. But anyway, you guys will be able to start to design drugs. I think maybe after our next class. I just want to check the chat real quick. Okay. Using AI. Ooh, good questions, people. Is polypharmacology a drug that can help with 2 different reasons, such as cholesterol or something else? Yeah, right. So sometimes it targets two complications. If that's definitely what you're asking, Madison, that's exactly what happens. Are researchers using AI at all to find optimized molecule? Oh, big time. You can't have a conversation without the AI. The AI or machine learning is also another big one. So those are huge. I mean, if you Google that, you'll find 1,000 papers on it. Are the statins considered Me Too drugs? Yeah, Madison, it's a popular question. It's like how many do we need? I would say yes, but they're an effective one. So they are sort of allowed to continue to produce them. People create sort of new reasons for using them. That's the way you sort of get around that. But if you create a Me Too drug that sort of does the same thing on one particular target, but then you're like, hey, wait, it can still, it does that, yes, but it also, I don't know, doesn't give you as much of an effect in terms of, either raising or lowering cholesterol, content, for example, then or bioavailability is another sort of big selling point. Then it sort of can sneak through. But usually it's almost in sort of an established situation. Like Merck was able to sort of, create the one drug and then sort of, pivot over to the next one. Usually once you sort of achieve the space, you can then continue to develop in that area. Where's the serine? Whew. That's a, it's a broad question. Did I miss it? All right, we can talk about that next time. I'm assuming that's in the slide. I'm sorry. It's hard to look at your question. Oh, okay. That's you know what? I'm going to start asking you guys that question. Maybe that's going to be an exam question next time. Okay. Thanks, Aaron. It's a funny one. Okay. Thank you guys so much for your attention, bearing with me on Zoom. I know a lot of you guys don't want to be on Zoom, so I get it. Next time, I'll let you know how I'm feeling tomorrow, and then we'll go from there. Thanks so much. Appreciate it.
Thank you, Dr. B.
Hey, thanks, man. Appreciate it. It's bad. Thanks, Dan. Thanks, Dave.